In this video we will discuss about soft matrices perceived more stem cell features. So from the literature they come to know that the matrices might also affect the stem cancer cells behaviors or stem cancer cell features. So they check the effect of the soft and stiff matrices on some of the genes that are involved in this stem cancer cell features. For example, they checked SOX2, c-KIT, OCT34, Nanog, CD133, nestin, BIM1, control is beta-actin. So they first checked it using reverse transcriptase PCR, and from that they are able to see that most of these genes are upregulated in the soft matrices compared to the stiff matrices. Thereafter, they also did real-time qPCR, and there they have seen the similar results, and the quantitative analysis also shows that the genes that are involved in the stem cancer cells are upregulated in the soft matrices compared to the stiff matrices. However, they also did these observations, or they checked these observations using another completely different approach, that is flow cytometer, and from that also the results are shown here for the two genes, that is CD133 and c-KIT. They are upregulated in the case of soft matrices compared to the stiff matrices. For example, the CD133 is upregulated to like 3.86 in the 1 kilopascal of the soft matrices compared to the 0.749 in the stiff matrices. And sorry, and for the case of c-KIT, they are able to see that upregulations is 4.87 in the soft matrices compared to the 2.36% in the stiff matrices. So this in all together indicates that from these two different approaches, it seems like the soft matrices promote more stem cancer cell features compared to the stiff matrices. And this is also in line with this previously published literature. However, these things seems to be counterintuitive in nature, that as the cancer cells become more aggressive in nature, their stiffness increases. So the cancer stem cells features goes down, and so that is one of the things that people need to understand, that when the cancer cells are in the onset stage, the stiffness is lower, so they are more closely toward these low stiffness features, and because of that they have more cancer stem cell features and they are able to replicate themselves. However, as they progresses, since the stiffness increases with the cancer cells progression, they are able to see cancer cells are more committed to a particular kind of behavior. And this is one of the reason that macrophages that comes in the tumor microenvironment, they then differentiate towards M2 type macrophages, which is more tumor proliferating in nature, whereas when it comes to the soft matrices, the differentiation is not that much. So this could be like one of the examples that support this idea that the soft matrices perceive more cancer stem cell features compared to the stiff matrices. One thing that they could have done of this flow cytometer results, they could have also shown the quantitative analysis of the flow cytometer results. So that would be like much more concrete evidence than just showing one representation of this flow cytometer. However, they did shows using these two different, not completely different approach. First one is reverse transcriptase PCR, and second one is real-time qPCR. And there they have seen the expression of stem cells related genes is significantly higher in the soft matrices compared to the stiff matrices. So I hope you understood how they or made a point that soft matrices perceive more cancer stem cell features. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.